Thanks everybody for joining us for this month's ERCC webinar. Um, I'm Roger Alexander, the Scientific Outreach Coordinator for the Extracellular RNA Communication Consortium. Today we have Juan Pablo Tosar with us. He's from the Universidad de la República in Uruguay and also the Institut Pasteur de Montevideo. He's an assistant professor there, and he's been doing sort of important work about the mechanism of release of non-vesicular xRNAs. He's showing that it's a selection process on xRNAs that are already in the extracellular milieu. What remains after digestion by RNases and other things in the extracellular space are the stable species, and rather than the sort of standard paradigm, which is selective secretion of specific species. So I may have oversimplified or misstated some of that, but we look forward to hearing the details from Juan. We do not have an October webinar scheduled yet, but in November, Michael Segal from MIT will speak to us. He's the first author of a recent science paper showing that a protein PEG10 encapsulates its own mRNA and can transit to target cells. And so they first verified that and then have been working to engineer it into an RNA therapeutic mechanism by altering the protein to carry different RNAs. So a lot of interesting work going on in the field, and we look forward, Juan, to hearing about their work.
Okay, thanks, Roger, and all the people involved in the ERCC2 outreach stream for the invitation to present in these webinars. I'm really happy to having the opportunity of sharing our work with you. So today we are going to talk about non-vesicular xRNAs. And just a disclaimer, this guy in the picture is Bruce Willis, that's not me. So let's start with what looks like a very basic question. What is the RNA composition of the extracellular space? We can say that this depends on three main variables. There are others as well. The biofluid that we are going to study, the type of xRNA carrier that will transport the RNA in that biofluid, and the analytical method involving our RNA extraction phase, the RNA sequencing, and data analysis. So if we have a look at the xRNA atlas, we will see that most of the studies submitted to that atlas are performed in plasma. So there's a lot of interest in plasma. There's an important difference between plasma and serum, for example. If we look at the RNA carrier, many studies have not done specific enrichment in specific carriers, so they have been done in total cell-free biofluid RNA. But then we have lots of studies performed looking specifically at extracellular vesicles or exosomes, so basically EVs, and some study performed in HDL and lipoproteins. But we do not see much studies looking specifically at non-vesicular, non-lipoprotein RNAs, which, by the way, is what we are going to talk about today. So this will not be a very popular talk. And there is most of the studies also perform small RNA sequencing, which requires a 5' phosphate and a 3' OH in the RNA. And this is great to study microRNAs and some no-coding RNA fragments. However, long RNAs or small RNAs having a 5' OH, a 3' phosphate, a 3' cyclid phosphate as a result, for example, of endonucleolytic cleavage by RNase family members, or having certain modified bases or being highly structured, like tRNAs, are not amenable to reverse transcription or amplification, so do not typically appear in sequencing performed with this type of methods. So from all the theoretical space involving extracellular RNAs, we can say that we mostly explore a part, and there's still more left to study. And most of our understanding actually comes from studies performed in cell culture, looking specifically at extracellular vesicles with an interest particularly in microRNAs. But let's not forget that the concept of extracellular RNA is broad and is not equivalent to EV-associated microRNAs. So in that case, we are talking about a specific carrier, which are extracellular vesicles, and a specific RNA biotype, which are microRNAs. However, if we look at the number of papers that were published last year with the word extracellular vesicle or exosome and microRNAs in the title or abstract, they account for 80% of the papers published last year talking about extracellular RNA. So there is a lot of interest which is justified in extracellular vesicle-associated microRNAs, but there are more stuff beyond that. So to illustrate the impact of the method, today the focus of this talk will be on tRNA fragments, so tRNA hubs and other tRNA-derived fragments that we will call TRFs. And I don't have time to introduce the biology of tRNA fragments, but let's just say that either in yeast, bacteria, or mammals, when the cells are exposed to stressful conditions, some anticodon nucleases are induced, and they cleave tRNAs by the anticodon, releasing tRNA fragments and tRNA hubs. And these tRNA hubs are a bona fide small RNA, so because they interact, they participate in different processes inside the cell. And like 6 years ago, we performed this study where we sequenced these small RNAs in MCF7 cells, and we compared that with small RNA sequencing in large EVs and in small EVs obtained by ultracentrifugation. It's pretty obvious that there is an enrichment in this population, shown in purple, which are tRNA fragments of 30-31 nucleotides in the small EVs, which compared to the cell are enriched in the small EVs. MicroRNAs are higher in the cells, but not so in the EVs, at least in our hands. However, some years later, the groups of Lambowitz and Sheckman published this study in EVs where they used Tiger-seq, which instead of using a conventional retrovirus derived reverse transcriptase, they use a thermostable group 2 intron reverse transcriptase, which is more amenable for the reverse transcription of highly structured RNAs, and is also less dependent on modified bases. And in this case, they observed that the major content of EVs were full-length tRNAs, not tRNA fragments. So here you can see two different sequencing methods telling completely different stories. And in fact, the Tiger-seq results are consistent with what we later observed by performing density gradient separation of cell conditioned medium and northern blot, where we see dense signal for full-length tRNAs in the vesicles, but not so tRNA fragments are below our detection limit. However, we see an accumulation of tRNA fragments outside and not inside the vesicles. So here we can see the impact of the sequencing method, and at least Tiger-seq seems to be more reliable in terms of the ratio between full-length tRNAs and tRNA fragments. Regarding the sample or the biofluid type, let's say that tRNA-derived fragments are high in serum, but low in plasma, as can be seen from this earlier study. However, the problem is not plasma itself, because if serum is treated with EDTA, then the signal of these tRNA halves is lost. And in contrast, if plasma is obtained with heparin as an anticoagulant instead of EDTA, then the signal of the tRNA halves is there. So if you want to study tRNA fragments, tRNA halves, then you need to preserve divalent cations in the medium. And this was confirmed later by Tom Tush's lab. Here the authors observed that the amount of microRNAs is lower in serum than in plasma. However, the total amount of RNA is higher in serum than in plasma. And what is lost between serum and plasma? Mostly tRNA fragments. And this has been confirmed by later high-throughput studies performing different biofluids. So these studies are not necessarily equivalent, but they all show the same pattern, that the amount of tRNA, the relative amount of tRNA fragments in urine is higher than in serum, and in serum tends to be always higher than in plasma. And this comes from one of the ERCC1 flagship papers. They also observed that, irrespectively of the biofluid type, tRNA fragments tend to be derived from glycine and glutamic acid tRNAs, and usually have a size between 30 and 35 nucleotides. So tRNA halves of glycine and glutamic acid cleaved at the anticodon loop. That's the most abundant tRNA fragments. Here I'm showing serum, but they observe the same in different biofluids. Regarding the type of xRNA carrier, I don't feel the need to introduce extracellular vesicles to this audience. Many of you are experts in this, but let's not forget that we also have extracellular RNA outside the vesicles, and we typically refer to the non-vesicular fraction as the ribonucleoprotein fraction, because we assume that the RNAs need to be protected by formation of complexes with proteins, otherwise they will be degraded by the action of extracellular ribonucleases. And in fact, that is the case for microRNAs. So these two seminal papers published 10 years ago show that the majority of microRNAs in human plasma do not coisolate with EVs. Some do, like LED7A, but most do not. Instead, they are forming complexes with the protein Argonaute2. And these AGO2 microRNA complexes are highly stable, at least in human plasma. And there are also reports showing the presence of extracellular RNA in lipoproteins like HDL. Interestingly, these tRNA hubs, which are abundant in biofluids, tend to not coisolate with vesicles. Here, this is another plot from the same study that I showed earlier, where the authors observed that these glycine tRNA hubs remain in ultracentrifugation supernatants in mouse serum. And we and others observed the same in cell conditioning medium, separating the vesicles from the ribonucleoproteins or the non-vesicular material, either by ultracentrifugation, filtration, or density gradient separation, as shown before. Here we are separating EVs and ribonucleoproteins by density, and we see that the tRNA hubs, especially glycine tRNA hubs, remain mostly outside EVs. So it is important to define which RNA biotype are we interested in studying beforehand in order to choose the adequate conditions to be able to retrieve and study those extracellular RNAs. For instance, if we are interested in full-length tRNAs, then we need to use Tiger-seq or tRNA-seq or some specific sequencing methods and look at the EVs. So we need to enrich in EVs if we want to study these tRNAs. In contrast, if we want to study tRNA fragments, then we can use standard small RNA sequencing because they tend to be amenable to standard small RNA sequencing, but they are more enriched outside EVs. So if we pre-enrich in vesicles, then that will not be so representative of the real population of these tRNA fragments. And the same, we need to go to serum. We cannot use EDTA or citrate-treated plasma. And why should we bother to study these longer and structured RNAs like tRNAs? Well, I think that an important answer is that they seem to be very abundant, at least in the vesicles. And there are a lot of great studies showing important biological effects induced by microRNAs in vesicles when they are taken up by cells. But let's consider that for every copy of a given microRNA that a cell is taking up through EVs, the same cell is probably taking more copies of tRNA hubs and maybe thousands of copies of full-length tRNAs, because that's what our abundance estimations show. And we really know very little about the biological effects induced by this abundant population. So we are trying to study that and trying to address the effects of tRNA hubs in the vesicles. To do that, we have been transfecting cells with synthetic labeled oligonucleotides with a sequence of some of these tRNA hubs. We see their encapsulation in vesicles by RT-qPCR, and if we expose acceptor cells to these vesicles, then we can see since these are labeled RNAs, we can see the signal in the acceptor cells. That depends a lot on the stability of these tRNA hubs, and we will talk about this soon. And we are currently in the process of analyzing some transcriptomic data that we have generated, but our intention is to move forward also to study the effects induced by full-length tRNAs containing modified bases if possible. So after this long introduction, let's go to what will be the focus of today's talk. We'll talk about non-vesicular RNA specifically, and I will show some studies performed in cell conditioned medium treated or not with ribonuclease inhibitor, given a method that we call RISE-seq And we'll show how full-length tRNAs and other full-length transcripts are converted into stable RNA fragments by the action of extracellular ribonucleases, introducing the concept of extracellular RNA metabolism. There are reports showing RNA processing inside vesicles. You might be familiarized with this study by Kalaluris lab, where the author showed that if vesicles contain the protein dicer, then pre-microRNAs encapsulated in the vesicles can be converted inside EVs into mature microRNAs. Not everyone has found dicer in the vesicles, so this is still an open topic. And there are also reports suggesting the processing of yRNA inside EVs as well. There are reports showing an enrichment of certain ribonucleases in the vesicles. So at least in theory, if a same vesicle encapsulates a ribonuclease and an RNA, that RNA can be processed in the vesicle. But in order to study that, we need to use like single vesicle analysis platforms. Some of them are being developed as part of ERCC2 efforts, as you know. And so I will try to tell the story of our voyage with extracellular RNA metabolism outside vesicles. I already shown these results where we saw an enrichment of tRNA hubs in small EVs, but I didn't show that we also sequence what's outside EVs, so what's left in 100,000 G supernatants, and in that case we see a huge enrichment of tRNA hubs, much higher than in vesicles. And in fact, most of the extracellular RNA in cell condition medium under serum-free conditions comes from this non-vesicular fraction. If we analyze the tRNA fragments inside the cells, we see that we have different fragments coming from different tRNAs, they have different lengths. If we sum up the reads from glycine and glutamic acid 5' hubs, they account for 45% of tRNA-derived reads. In contrast, in the non-vesicular fraction, they account for almost 90% of tRNA reads. So they are mostly glycine and glutamic acid fragments of 30 or 31 nucleotides. And at that time, we thought that this could be indicative of a selective secretion process. So the cells might be releasing these glycine and glutamic acid tRNA hubs. However, some years later, we realized that these glycine 5' hubs have something in particular, and it's that they can form these homodimers. And this is an in silico prediction of the secondary structure. They form the stem loop structure with an unstructured 3' tail, but that 3' tail is self-complementary. So at least in silico, we can predict and model these beautiful homodimers. And I call them beautiful because they really do not show many single-stranded bases for single-stranded endonucleases to clip them. So they look like resistant to endonucleolytic cleavage, and the extremes are also buried inside the structure, so they could also be resistant to exonucleases as well. Working with synthetic oligonucleotides, we showed that they elute as dimers in size exclusion chromatography, but if we introduce a mutation here, a change here, in order to disrupt the putative dimerization interface, we just change one base, now they eluted as monomers. And we got a linear relationship between the concentration of the dimer and the square of the concentration of the monomer, as expected for a monomer-dimer equilibrium. If we transfected these oligonucleotides, labeled oligonucleotides with biotin at 5', inside cells, we observed that after 6 hours, if we transfect with a scrambled oligonucleotide or with these mutants that cannot dimerize, they disappear from cells in less than 6 hours, so they are rapidly degraded. In contrast, if we transfect with the wild-type sequences, or with this second mutant, where we change 2 guanines here for 2 adenines, so this is a mutant that can dimerize, these RNAs remain in the cell. So this shows that there is a correlation between the capacity of forming dimers and the stability inside cells, and we also show the same for in vitro assays treating with endonucleases and exonucleases. So these oligos are really more resistant to degradation. So this is what we observed by our sequencing study. We observed an enrichment of glycine and glutamic acid tRNA hubs, fragments of 30, 31 nucleotides outside the cells compared to inside the cells. And this could be indicative of a selective release mechanism. However, there is an alternative explanation, and is that cells might be releasing these fragments following the concentration that they have inside the cells, but then extracellular ribonucleases could cleave and degrade most of these non-vesicular fragments, and those that will remain are those that show a higher stability, maybe because of their capacity of forming dimers, maybe because they just bound a protein in the extracellular space and they are stabilized that way. So to distinguish between these two scenarios, we decided to add ribonuclease inhibitor, or RI, to the media and see if we could freeze this situation over here. So here I'm showing a chromatogram after SEC analysis of MCF7 cell conditioned medium after ultracentrifugation. So we are injecting the supernatant, the non-vesicular fraction, and we are seeing this peak at 260. So this is indicative of the elution of nucleic acids. If we treat with RNAs before SEC, that peak is lost. This means that this is an RNA peak. And if we treat the medium with RI, with ribonuclease inhibitor, then the intensity of this peak that we call P1 is increased. We run the RNA in this P1 peak in a denaturing gel and observe that when RI is not present, then we have mostly fragments of 30 nucleotides. However, if we add RI to the medium, then we stabilize full-length tRNAs. We perform small RNA sequencing in this P1 peak and observe a difference in the distribution in the length of these fragments. This can be inferred from the gel as well. So our interpretation is that because standard small RNA sequencing does not capture full-length tRNAs, what we are capturing are the 5' tRNA halves that are cleaved at the anticodon, which is like the primary cleavage site for tRNAs. However, when RI is not present, then we are only capturing those fragments which show the higher stability, and those are the fragments of 30 nucleotides, presumably because of the capacity of forming dimers. If this is correct, we should show that the fragments of 30 are much more stable than the fragments of 33, 34, 35. And to do that, we incubated the cells with serum-free medium for just 30 seconds, so a very brief incubation, but some RNAs are released. And then we incubated that cell conditioned medium at 37 degrees for different periods of time. And you can see that this really looks like a cell lysate. So the RNAs are released to the extracellular space as they are in the cells. We can see full-length tRNAs by northern blot. And after a cell-free incubation at 37 degrees, we see a slight increase in the intensity of these fragments, these tRNA halves of 34 nucleotides. However, if we incubate in the presence of serum for just 1 hour, so serum is a rich source of ribonucleases, then we completely lose these full-length tRNAs. The fragments of 34 also tend to disappear, and instead we have an accumulation of the fragments of 30 and 31. So this kind of proves that these fragments of 30 are really, really stable in the presence of serum. And then we just analyze standard cell conditioned mediums, or 24 hours in the presence of DMIM plus 10% FBS. And I already showed part of this analysis, and we separated the vesicular from the non-vesicular fraction by flotation gradient in iodic channel gradients following established ERCC1 protocols. And as mentioned earlier, we see the presence of full-length tRNAs in the vesicles. We do not see the full-length tRNAs outside the EVs. This is in the presence of serum, but we see the accumulation of the fragments of glycine of exactly 30 nucleotides. So what we propose is that cells release full-length tRNAs to the extracellular space and outside vesicles, but we typically do not see them because they are rapidly degraded by extracellular ribonucleases, forming fragments of 34, which are also degraded, and fragments of 30, which accumulate, and those are the ones that we can sequence. In contrast, when cells release full-length tRNAs inside vesicles, these remain stable and can be seen in the EVs because they are protected by the action of extracellular ribonucleases, because the RNAs cannot enter in the EVs. So while our manuscript was still under review, this second paper was published by the group of Tom Chicheras at Cold Spring Harbor. They were submitted mostly in the same date, but their study was published earlier, and I was really happy and excited to read it because it showed pretty much the same concept following a completely different approach. In this case, the authors use a leukemia cell line and they use CRISPR-Cas9 to remove the RNase 1 gene from these cells. And they grew the cells in serum-free medium. And what they observed when analyzing the non-vesicular fraction was the presence of fragments of glycine GCC of 31 or 30 nucleotides. However, when the cells lacked RNase 1, then they observed full-length tRNAs. So either in our study, inhibiting the action of extracellular ribonucleases, or in their study where they remove an enzyme from the cells, so the cells can no longer secrete the RNase to the extracellular space, we can see that this dynamic process is going on. So this gave us confidence that this is what's happening, even if we compare small RNA sequencing inside cells and in extracellular samples, we will see an enrichment of glycine GCC fibrine hubs, shown here in red. In the extracellular fractions, we can think that that's a consequence of selective secretion, but actually it's a consequence of extracellular biogenesis of these tRNA fragments coupled to differential RNA stability. So cells are probably releasing full-length tRNAs, and those tRNAs are cleaved in the extracellular space by extracellular ribonucleases. Many of the fragments are degraded, but some accumulate because of their high stability. So we wrote about this in a perspective, in a recent perspective that we published with Ken Whitwer and Alfonso Cayota, and we suggest that the survivorship bias is what defines non-vesicular xRNA profiles. And if you write survivorship bias in the internet, you will probably see these diagrams representing US bombers when they return to the base after a mission in World War II, and people look at the places where they had bullet holes, and the statistician Abraham Wall suggested adding extra armor in the places where holes were never seen, and that could seem counterintuitive, but those were the places that when the planes were hitting those positions, they could not return to the base. And another way of seeing survivorship bias is like by reading the biographies of very accomplished and successful people, like Steve Jobs, who dropped out of college very early and was very successful, but we are not reading the biographies of all the people who left college early and were not. Oh, there was a second very obvious and interesting observation when we added ribonuclease inhibitor to the media, and it's the presence of this peak that we call P0 that now appears with a lot of absorbance at 260. And we analyzed the RNA in this peak that we call P0, and we observed that it was composed of high molecular weight RNA. When we performed small RNA sequencing in that peak, we observed a lot of ribosomal RNA fragments. But of course, small RNA sequencing is not capable of sequencing full-length ribosomal RNAs, which are very large. But there is one, well, actually there are two that are short. One of them is the 5.8S ribosomal RNA, and in this case, we had reads covering the entire 5.8S ribosomal RNA. So that gave us confidence that we could be having full-length ribosomal RNA in that peak. And to study that further, we took the RNA in this P0 peak and reverse transcribed it with random primers and used different primer sets spanning different regions of different ribosomal RNAs, and we could observe that in all the cases we could amplify them almost with the same initial concentration in the P0 peak. So this is indicative of the presence of not fragments as our small RNA sequencing suggested, but of full-length ribosomal RNAs. Do we have ribosomal proteins? So we did mass spec in this P0 peak, and here in red, each dot represents our ribosomal protein. They are mostly all there in the P0 peak. However, if we treated the cell condition medium with RNAs before SEC, then the presence of all this protein was lost, as can be seen here. So this means that ribosomal proteins are forming a high molecular weight aggregate that elutes in the exclusion volume of the columns, and that aggregate is dependent on RNA. And we know that we had full-length ribosomal RNA, so we reasoned that we could have ribosomal subunits or maybe ribosomes, and we have some TEM negative staining that suggested that that could be the case. And it's known since the 60s that if you treat ribosomes with ETTA, they will disassemble, so the subunits will be separated. So we incubated the cells with medium lacking calcium or magnesium in order to have ribosomal subunits disassembled. And then we use differential ultracentrifugation. So there are no vesicles here, but we are using differential ultracentrifugation to try to collect the subunits by centrifugation. And we need to go to 100,000 G in order to pellet the ribosomal RNA. In contrast, if we incubated the cells with a medium containing calcium and magnesium, we could pellet the ribosomal RNA at a much lower speed, suggesting again that maybe we're having not just ribosomal subunits, but full ribosomes. And to study that further, we follow a protocol which is typically used for polysome profiling, but instead of using a cell lysate, here we were using concentrated cell conditioned medium treated with ribonuclease inhibitor. And basically what we see in the upper fractions of the gradient is the presence of tRNAs and free proteins, but then we see a peak at 260 where we see the 18S ribosomal RNA and proteins coming from the small ribosomal subunit by Western blot, but not from the large ribosomal subunit. And then this higher peak, here we can find all the ribosomal RNAs and proteins from both the large and small ribosomal subunits. So this could be monosomes, ribosomes. And then these patterns kind of repeat itself. So we have like maybe 2 ribosomes, and this could represent 3 or more. But the signal here for the proteins is very weak. So we pre-treated cells with cyclohexamide, which freezes polysomes inside the cells before incubating them in the medium and repeating this analysis. And by doing so, we could stabilize the signal in these deeper fractions of the gradient. So this suggests the presence of ribosomal aggregates or maybe polysomes. If we have polysomes, then we need to have mRNA, full-length mRNA, because polysomes are constructed on top of mRNA. So we went back to our small RNA sequencing data in the P0 peak. We observed reads for fragments of different mRNAs, but to distinguish whether we had fragments of full-length mRNAs, we reverse transcribed the RNA in this P0 peak with an oligo-DT, and then amplified with primers at the 5' end of those mRNAs, obtaining single bands of the expected size. And to further show the presence of intact mRNAs, we use a nanopore direct RNA sequencing of RI-treated cell conditioned medium. And we obtain like a really low number of reads, to be honest, but these are long reads and they span, each read has the length of an entire mRNA. So looks like we also have ribosomes and mRNAs. So we asked whether these extracellular ribosomes could be functional, and to that we added puromycin. Puromycin is a drug, it's an antibiotic which mimics a tRNA coupled to the amino acid tyrosine. Puromycin can enter in the A site of the ribosome, and if there is a nascent peptide in a P site, it will be transferred to puromycin, and translation will be stopped. And we can detect the puromylization of peptides with an anti-puromycin antibody. And we did that with concentrated extracellular fractions, incubated at 37 degrees in the presence of puromycin, and we see a signal suggesting that we had some puromycin incorporation, but less so at 4 degrees. However, this was not dependent of ATP or GTP. So the peptide transference reaction is thought to be an ATP-independent process, but elongation is not. It requires GTP. So there's no evidence for elongation, but to the best of our knowledge, this is the first evidence of the peptide transferase reaction occurring in extracellular samples obtained without deliberated cell lysis steps. And I think that the word deliberated is key here, because we were really trying hard not to lyse or kill the cells in any way possible by doing these short incubations with medium, but we can never discard that a very little number of cells will die and release their contents, and in fact that's what we think that is going on. But if that's the case, this will be the same for any studies performed in cell culture regarding extracellular RNAs. So this is the image that we suggested for the cover of NAR, and hopefully and was accepted. And here we are represented a ribosome and some tRNAs in the extracellular space. Here you can see a monolayers of cells in the back, and the action of extracellular ribonucleases represented as scissors, degrading these ribosomes and these tRNAs and generating some fragments which remain and accumulate in the extracellular samples because of their differential stability. So this is our current working model. This is what we are like, how we see the biological relevance of all this. Let's imagine a tumor microenvironment or a cellular microenvironment where cells are releasing EVs containing full-length tRNAs. So we have talked of them earlier. Stress cells could also be inducing the packaging of tRNA hubs and other stress-induced tRNA fragments, but we already talked about that earlier. But now we also see the release of full-length tRNAs and ribosomes, maybe from dead cells, maybe as an active process from lived cells. We are still not sure about it. But what's important is that these ribosomes and tRNAs will not travel far from the place where they are released because of the action of extracellular ribonucleases that will degrade them. However, immune cells or other cells in close proximity will still sense and react to this extracellular non-vesicular RNA. And in fact, we have some evidence that that's the case, and Mauricio Castellano is working on the immunological aspects of these processes. But what's important is that the degradation of these ribosomes and tRNAs left a byproduct, which are these very stable tRNA fragments and some very stable ribosomal RNA fragments that might be able to diffuse for longer distances and maybe interact with other cells. And Marco Licalzi is doing his PhD studying this. So it's important to know the half-life of these non-vesicular RNAs in different extracellular conditions, in different biofluids, because there is an inverse correlation between degradation and diffusion. So the capacity of these non-vesicular RNAs to reach longer distances from the place where they are generated. And Bruno Costa is working on that project. So this is what we are currently doing as part of our contribution to ERCC2 efforts. We are purifying RNAs and ribonucleoproteins from cell lysates, and we are incubating them in different biofluids, urine, serum, CSF, for increasing periods of time at 37 degrees. And we are measuring the half-life of full-length transcripts and their fragments by northern blot. What for? We want to use this information to identify novel RNA fragments with high resistance to extracellular ribonucleases and design specific assays to measure them in biofluids. So we see this as a different approach for the discovery of RNA biomarkers, of extracellular RNA biomarkers. Instead of using standard sequencing methods to compare patients against controls, we will look for RNAs that are highly stable in biofluids, but that are typically not recovered by standard techniques, and in order to design specific assays in order to measure them and see whether they change or not in patients of certain disease against controls. And I think that it's always nice to show some unpublished data of the work that we have that we are currently doing. This is what happens when we spike purified RNA in 10% fetal bovine serum, but as I said, this was repeated in pure serum and in CSF and in other biofluids, and we are also spiking ribosomes, purified ribosomes. And by northern blot, with a probe against the 28S ribosomal RNA, we see that when we add purified RNA, here you can see with RI, with RNase inhibitor, we can stabilize the ribosomal RNA, but it is rapidly degraded in less than a minute when added to serum. In contrast, when we add ribosomes, we can detect the full-length 28S ribosomal RNA, and we can measure its half-life in 10% serum, not only of the full-length transcript, but also of some fragments which are generated. So by doing this, we can measure different half-lives. And something which is interesting is that although 28 days ribosomal RNA is rapidly cleared from FBS. That's not the case for every transcript. Like here, you can see a signal which comes from the place where we expect tRNAs, and by Northern blot against a tRNA, in this case lysine, we can see that the lysine tRNA remains in FBS 10% for at least actually 90 minutes. In contrast, glycine GCC is degraded in less than a minute. So there are striking differences not only between different RNA biotypes, but also among the same RNA population like tRNAs. And this is actually not new. We have already observed that with the endogenous with endogenous full-length tRNAs that are released by the cells to the medium. In this case, we incubated serum-free cell conditioned medium with Array, obtained with Array for 24 hours in the absence of cells at 37 degrees. So we know that we have all the tRNAs in that sample initially, but after 24 hours, we see lysine UUU there, but we do not see glycine GCC. And the fragments are inversely correlated in the stability with the full-length transcripts. So Northern blot analysis showed marked differences in tRNA stability, with lysine being highly stable, glycine not so much. And we have already published that, but we are observing the same when spiking the RNA in the biofluid directly. And we think that modified bases can explain this phenomenon because lysine UUU contains some very bulky modifications in the anticodon. So this will probably make this tRNA less prone to degradation. So what we have learned by doing this type of studies, we typically say that extracellular RNA needs to be protected from extracellular ribonucleases by their encapsulation in vesicles, lipoprotein particles, or by formation of ribonucleoprotein complexes. That is true for ribosomal RNA because the stability of ribosomal RNA in ribosomes seems to be much higher than the stability of naked ribosomal RNA, and that's not surprising. However, naked tRNAs are quite more stable. Some of them, like lysine, are very stable, with relative half-lives in pure CSF of more than 2 minutes, which is quite, it's not bad. The presence of modified nucleobases probably drives differential stability among tRNAs, and some tRNA fragments are extremely stable, with relative half-life in pure CSF of more than 2 hours. So that's a lot. And thus factors affecting the stability of non-vesicular xRNAs are protein binding, post-transcriptional modifications, and RNA structure. All the three factors influence the stability of any given RNA in the extracellular space, when of course not protected inside EVs. So as conclusions or take-home messages, although many of you might be connected from home, so not necessarily take-home messages, not all potentially interesting xRNA biotypes are found in a given biofluid. Take the case of serum versus plasma regarding tRNA fragments. Not all potentially interesting xRNA biotypes are found in EVs. Most sequencing techniques are targeted analytical techniques. Small RNA-seq is great to study microRNAs and some tRNA fragments, but other xRNAs are also present and they seem to be more abundant, even if they are not captured nicely by these techniques. Survivorship bias defines the extracellular RNAome, not a problem for biomarker studies, where in fact we want to study things that are stable and they resist degradation, so we can find them in archived cohorts. But we need to look at both stable and unstable extracellular RNAs to understand RNA release mechanisms. Non-vesicular extracellular RNAs can be short-lived, but this does not discard low-range paracrine effects. However, some non-vesicular xRNAs are highly stable, and they are detectable in biofluids with high RNase activity. Talking about non-vesicular xRNAs is not necessarily a synonym of extracellular ribonucleoproteins, because stable naked xRNAs are at least feasible. Structure coupled to modified bases can do the trick of stabilizing certain fragments or transcripts. So the concept of non-vesicular xRNA metabolism is composed of two concepts. We need extracellular biogenesis, and I hoped to have provided some competing evidence showing that this could be the case at least for tRNA hubs in the non-vesicular space. coupled to cell uptake and downstream effects, something that we are working on right now, and hopefully in a future talk I can show some specific data on that. So to finish, I would just like to acknowledge and thank our fantastic team. We are actually a merged lab between my group at the School of Science of Universidad de la República and Alfonso Cayota's lab at the Pasteur Institute of Montevideo. So we kind of merged both groups in order to work in these RNA projects. And I would also like to thank Ken Whitwer, with whom we have collaborated a lot in the past, and we are part of the same ERCC2 team, which is led by Ken. Part of the work I've shown was done at Pavel Ivanov's lab at Brigham and Women's Hospital in Boston, where I stayed in his lab for some time recently as a visiting scholar, and the dimerization work was done in collaboration with Eric Westhoff. And we have also been working in extracellular piRNAs that I didn't have time to show any data on that, in collaboration with Carlos Rovira, a fantastic scientist. I learned a lot from him, and he sadly passed away recently. So thanks a lot, and well, I'd be happy to take your question and continue discussing with you.
Juan, thanks very much. That's a lot to digest. I would ask people if they have questions to unmute themselves, introduce themselves, ask their questions, and then mute again so we don't have too much audio feedback. Here's a question in the chat from Joe, who asks, do you also study circular RNAs in biofluids or conditioned medium, as these are presumed to be resistant against RNases?
Yeah, not yet, but after reading their paper, we definitely are thinking in starting to study that. And the fact that they observed an enrichment in circular RNAs in biofluids, I think it's suggestive of that, again, the differential stability might be playing an important role there. But we haven't done that yet.
I would follow that up by asking you about that recent Cell paper showing RNAs expressed on the cell surface protected by glycosylation. What are your thoughts on that?
Well, we have had some discussion in the past with Ryan Flynn, who's the first author of that paper. And I think that, well, it's obviously a very important work and mind-blowing in many senses. But is it that the cells are exporting the RNA from the inside and exposing the RNA in the surface? What's that contribution compared to the possibility of extracellular non-vesicular RNA, which might be glycosylated, and glycosylation might explain the stability of some of these non-vesicular RNAs, whether those non-vesicular RNAs could stick to the surface of the cells. I don't know if that has been like resolved in that study yet. So both things could be contributing to that phenomena, but of course it's really interesting.
Yeah, I don't think that has, obviously it's a single study and there's a lot of work left to do. I don't think that has been clarified, and to me it shows the importance of your work, because prior to your work showing, hey, we need to look at selection within the medium for stable species, people would have started, and they do need to start looking for, export mechanisms for glycosylated RNA, but it's important to consider the other mechanism that you point out.
Yeah, I agree.
Let me ask you, what is known about why do healthy cells release tRNAs in vesicles and non-vesicularly? What are they doing? Why are they doing that?
Yeah, tRNAs are very abundant in the cells. And I need to go back quite a lot. But we see an enrichment of tRNAs in vesicles here. Here you can see this is cytocol staining. We have vesicular fractions in here, and we mostly see bands corresponding to tRNAs, and we confirmed that by Northern blot, and also 7SL RNA. So the RNAs which are highly abundant in the cell, we tend to see them in the vesicles, but not the ribosomal RNA. So but probably that's because the ribosomal RNA, which is very abundant, is forming ribosomes, and the ribosomes are somehow excluded from this selection process. So there might be, and again, there's a paper from Randy Schechtman's lab suggesting that IBX1 is responsible of the export of tRNAs, or is involved at least in the export of tRNA into vesicles. But actually, if you have something which is very abundant in the cytoplasm, maybe you do not necessarily need an active process to take the RNA there. You just need some proteins you need to have the RNA, of course, in the site of biogenesis. So I'm not sure. There are reports suggesting the involvement of IBX1 in this case, but again, these are highly stable RNAs. So that's what I can say regarding the encapsulation of tRNAs in the vesicles. And regarding the presence of tRNAs outside vesicles, there are some mechanisms which at least are compatible with the possibility of thinking in a release of tRNAs and even ribosomes, which are very big, from healthy cells. And one of the ERCC flagship papers showed that, for instance, when an autophagosome is formed, it does not necessarily fuse, and that has been shown by other studies as well, does not necessarily fuse with lysosomes for degradation purposes, as we typically think in autophagy, but they can also fuse with the plasma membrane, and that could be a way of releasing large components, and you can actually release even organelles that way. But that could be a mechanism of releasing like cytoplasmic soluble complexes. Then there are other mechanisms involving cytoplasmic extrusion, which have been described. But there's also the possibility that since we are incubating the cells with serum-free media, which doesn't contain proteins, it has glucose, it has divalent ions, it has lots of things, and we are performing very short incubations, but maybe some cells which are more fragile will just explode in those brief incubations. And we look at the cells, and we even take pictures before and after, and most of the cells remain there as expected, but only a small percentage of cells dying and releasing their entire cytoplasmic contents into the extracellular space can outnumber what is being released actively from cells. So I think that's another technical challenge that we will need to solve in how to distinguish between both processes. Anyway, even if considering that non-vesicular RNAs is coming from dead cells, which I honestly don't know if that's the case, that's interesting from an immunological perspective, because we think that in tumors, for example, where there's also always an inner mass of necrotic cells, and we know that immune cells are part of the tumor, and there is a crosstalk between the cancer cells and immune cells. They might be sensing this extracellular RNA, and that's something that we are working at. So I don't think I have answered your question, because I don't think we really have the answer of what are the mechanisms involved explaining all this. It's still a lot of work to do in that sense.
I guess I would say two things. One is Steve Gould is always arguing that a lot more of the cargo of vesicles and the sort of vesiculation process is due to, is not selected but random, so that you would expect RNA species that are abundant in the cell to be abundant in vesicles. And to follow up, in a tumor microenvironment or in an experiment, when just a few cells lyse and you get their entire contents, I believe you said that for the full ribosomes, you believe that that's what's going on. And my question is, are the amount of tRNAs that released by just a few cells lysing enough to account for the high level of tRNAs that we see in, RNA-seq experiments?
Yeah, again, I don't
It needs
think
to be more carefully
I can give
studied,
a precise quantitative answer to that question, but we have measured the amount of extracellular RNA that we are capturing from the non-vesicular fraction after performing these very brief washes, and we sometimes did more than one in order to try to get rid of dead cells initially and then explore what is being released in just that very brief period of time. And there's still quite a lot of RNA in there. And we calculated like how many cells, if they were releasing the 100% of their cytoplasmic content to the extracellular space, how many cells would be needed to explain that amount of extracellular RNA. And our calculations are like 0.5 or even less of the culture. So even a very low number of cells being passively dying and releasing their content might explain part of what we are seeing. But of course, we are interested in knowing whether there is also some active source of release of at least the tRNA stuff. Yeah, but we are still not sure about it.
Yeah, I mean, it just goes to show how tricky the field is, and the careful experiments you're doing, there's still things at the edges that can't be clarified. Joe asks another question. Can you talk again about the difference between plasma and serum regarding small RNAs?
Yes. So I'm not an expert on that, and most of the work that I've shown is actually coming from studies performed by other people. So I will just talk about this as a reader. But I don't know whether he was referring to this, but for example, regarding tRNA fragments, glycine tRNA hubs, which seem to be abundant in certain biofluids, especially in urine, for example, the majority of tRNA reads, at least in this study, were tRNA fragments. That's not necessarily the case in other studies, which were also performed in urine, but they seem to be there at least. And in this case, the tRNA hubs can be detected in serum, and they can be detected in plasma, but if heparin is used as an anticoagulant. So the problem is not plasma itself. The problem is the anticoagulant, the choice of the anticoagulant. And typically plasma is collected with EDTA or citrate or divalent ion chelators. So divalent ion chelators will remove magnesium and calcium, and somehow the stability of these tRNA hubs or some of these tRNA fragments depend on them. So here with heparin-treated plasma, the tRNA hubs are there, they can be detected by northern blot, and with EDTA plasma, they are not. And if EDTA is added to serum, then the signal is partially lost. And well, other studies have compared serum versus plasma, and the population that is mostly affected are tRNA fragments. So they seem to rely a lot on divalent ions. And since most tRNA fragments, we know that they are not associated with vesicles, that makes sense, because even if they might be forming dimers or they might be forming complexes with protein, in any case, divalent ions are known to be important for that type of interactions. I don't know if I'm answering the question in the right direction.
I think so. It just goes to the point of experimental conditions are key to understanding what you find in the biofluids. I think we've run out of time. I have a half an hour of further questions, but we'll let everybody go. And thank you very much again, Juan, for this really interesting work. We'll be looking forward to seeing the follow-up work from your team, from your grad students. Thanks very much.
Thank you, Roger, and thanks a lot for the invitation. Bye, everyone.